Good morning, everyone. I am welcome you all to this 29th day of CSIR online training. Today we are going to discuss about Unit 12, Applied Biology. Today we are going. I will cover some small portions of the Unit 12. Before entering into the session, please let me know, am I audible? Please type it in chat box. Am I audible? Can you see my slides? Is it visible? Please let me know through chat box. Thank you for your cooperation. Sorry for the delay. Today I'm delay. Please, I'm going to start this session. Okay, today our section is unit third session of our planning. This unit mainly focused on the 1 mark questions and the conceptual based question. In the applied biology, we learned many basic concepts, right? That many microbiology, about cells, about plant, all those things we about the tissues in earlier and using that technology, based on DNA information, RNA information, all those things we discussed in previous classes. Okay, using that basic concept, we can apply in that it's a for a use, it's a for human life, that kind of thing. First is a fermentation process. Okay, the fermentation is a most widely used application. Okay, in day-to-day life we are facing the fermentation in normal curd production, and apart from that, many industries are producing the fermentation products. Why I am included this ethanol based first one means, today in current scenario, many other food industries also started this ethanol production, because it's a very needed one, so they have started this ethanol production. Ethanol can produce by the Saccharomyces cerevisiae. It produce the beverage and also the industrial solvents. In the industrial solvents, here they are using the sanitizers for production. They are produce the ethanol using the Saccharomyces cerevisiae. In this I am included some of the microbes alone. Apart from this, we have n number of microbes that I'll separately I'm having the table. In glycerol, that is the source organism is that Saccharomyces cerevisiae. It's producing the helps for the explosives. And lactic acid, that we are all know, Lactobacillus bulgarius, it's a food and pharmaceutical application. Here we want to know one thing. Normal all of the lactic acid Lactobacillus species are not able to produce the lactic acid. Some lactobacillus are harmful for the human system. So Lactobacillus bulgarius is a well studied and accepted by the FDA for the food and pharmaceutical production. And acetone and butanol, it's this acetone and butanols are produced by the Plasmodium acetobutylium. It's helpful for the solvent production. Alpha amylase, Bacillus subtilis, it's a starch hydrolysis. This alpha amylase is helpful for the starch hydrolysis. Okay, in this, how they are producing this solvents. Okay, that is very important thing. The microbes can grow in a medium which contains substrate. Okay, if there is a particular condition, higher glucose condition, for ethanol I am telling example, if there is a higher glucose concentration in a medium, that microbe utilizing the glucose, the carbon source, and producing the ethanol, because there is a lack of nitrogen. It's a higher quantity of glucose that activating the glycolysis pathway in a microbe. That pathway some pathway, it's the microbes having different pathways. The pathway activation, it's helpful for the production of bioethanol. Some products of the fermentations are produced by the external surface receptors of the microbe. That are in that case we can use the continuous cultivation. Okay, it's very important thing. Using the continuous cultivation means, here the two fermentation methods are there: batch fermentation and continuous fermentation. In the batch fermentation, means once you added the substrate and added the microbe, then taken the product, that set of experiment finished at that end. So in next step you need to add new substrate, new microbe, other new process. It's a cost, highly cost, highly expensive one, because you need to add a substrate in each and every step. That process, cleaning process in the fermentation, it's a most important step in many industries. They are using many thousand liters of water for one fermentation process. That is very demanded, so only they are taken many cost for some drugs, production of some drugs and fermentation case. The continuous cultivation means, if the microbe producing the product through the extracellular protein, means that extracellular protein utilizing the substrate and producing the product in a medium. So if we are taken the medium and we can medium in to the next step and add the another medium. It's a continuous process. These are all the some important things in the fermentation process. Okay, lactic acid production. In the lactic acid production, we can see it's a normal chemical synthesis method. It's a microbial method. In the microbial method, the biomass, we can take the biomass. That biomass is our designed one. We can't take biomass here, the pre-treated enzyme hydrolyzed biomass. That may be the carbohydrate, fermentable carbohydrate, that is the polysaccharides. This fermentable carbohydrates are added taken in a medium, then add the microbes in this step. After the microbes addition, fermented cultures, we can get the fermented culture, then separate and purify the product. This is the optically pure, the lactic acid plus or minus we can get from the microbial cultivation. Okay, some microbes, this is the first one example. Here you can see the microbes, the E. coli and other bacteria helpful for the production of lactic acid, glyceric acid, PHP, polyhydroxybutyrate, dihydroacetone. In this for example, lactic acid, we all know lactobacillus, that is a bacteria. PHP, Bacillus subtilis species are produced. They can easily produce that PHP product. In this case, first I told ethanol production, we need a higher quantity of glucose in a medium. In this PHP production process, we need higher quantity of nitrogen source in a medium. Nitrogen source, that they are absorbed and stored on the cell surface, that is in lipid form. Okay, So that cells are uptake that nitrogen source and store in the cell surface, cell wall, in the cell wall. Okay, in the cell wall, not a cell surface, in the cell wall. Once yet that php, that stored lipids are so it's make us cell, it's they are helpful for their cell. Okay, after some period, if we need a fixed time for fermentation, don't allow the maximum time. If it may take 3 days or 4 days. After that particular 4 days alone, okay, first 3 days the microbes balancedly taken the carbon, hydrogen, nitrogen. After some particular period, in php, after the third day, it taken the remaining nitrogen source for their survival. So the nitrogens are stored in the cell wall. Okay, how we can confirm means the Nile red, if you are adding the Nile red or Nile blue over the cell. Okay, we want to take the cell from the medium and add some nitric Nile red or Nile blue, and the fluorescence means that that gel dark machine or any fluorescence indicator means you can get the not fluorescent, sorry, that UV, under UV light you can get that illuminations, that color change, that differently fluorescent, that differently coloring the cell wall. It looks with the green color or red color zones. Okay, it's a difference, that is, it is indications. And the yeast and filamentous fungi in aerobic condition, they are producing the oxalic acid, citric acid, mannitol, erythritol, arbitol. This are all in aerobic condition production. Okay, the very important thing, succinate and these are all oxalic acid, citric acid, these are all the things are able to produce in aerobic condition. In enterobacterios, that is an anaerobic condition. In the anaerobic condition, we can produce 2,3-butanol, butanediol and butanol. In this case, we can produce 1,3-propanediol. Okay, and ethanol. Ethanol it can be producible by E. coli, bacteria, yeast, and enterobacteria. The many things. This as per the research reports, bacteria are maximum producing the this ethanol. Saccharomyces are widely used because that's user friendly. Okay, this some natural methods, it's a method pathway, because very few things, because petrochemicals are, it's a chemical process that it's not it in upcoming 100 or 200 year it may exit, because there was lack of the source, so we need alternative for our for the natural resource. Here the petrochemical resource we can taken, that is the renewable, that they are added the that are pre-treatment means the acid hydrolysis. The pre-treatment step in the fermentation is very very important. The pre-treatment means substrate preparation. The substrate preparation, they added the acid, they did acid hydrolysis or enzyme saccarification. This acid hydrolysis means this acid break down the hydrogen bond between the polysaccharides, because every process without carbohydrate we can't do any experiment. In that case, we need to add acid to hydrolysis the and break down the polysaccharides, because that animals, that microbes can't able to take the whole polysaccharides. Like our human, we need to chop the vegetables and we are taking, right, that same process for the microbes we need to chop the polysaccharides through acid hydrolysis and enzyme saccharification. And that after the enzyme saccharification, that is carbohydrate that is able to taken by the microbe. This microbial fermentations happen, and the recovery and purification is producing the lactic acid. This is the here you can see we can directly go for the fermentation process with the simultaneous. It's a sideway process. It's a saccharification and fermentation. It's a process happen. And this it's a chemical method, it's a biological method. It's for the lactic acid production. Why this is the one alternative method for the lactic acid production. And in this fermentation pathway is very important one in the CSIR exam point of view, because we are asking the 2-mark questions, right? So in the 2-mark questions point of, so in the 2-mark direct one question in the part B question, okay, the part B, it's carrying 35. We need to attend the 52 marks. In that 35 is they are provided 52 mark. We need to answer the 35 questions. Here the first thing, always the pyruvic acid play a major role in the because you are given a glucose source, right? Every substrate, every medium having been converted by the glycolysis pathway and producing the pyruvic acid. Okay, in that pyruvic acid is if the pyruvic acid is utilized by the streptococcus, lactobacillus, staphylococcus, lactobacillus. These 3 microbes are utilizing the pyruvic acid. They can produce the lactic acid. Okay, then here E. coli and Salmonella. This E. coli and Salmonella species are helpful for the production of oxalolactic acid. Okay, and this is common for both. It's acetic acid. Okay, this is acetic acid. Acetic acid, it's produced by E. coli and Salmonella. Before that acetic acid itself, we need the formic acid. The formic acid is produced by Shigella. Okay, then the propionic acid, bifidobacterium and propionin bacterium, these are producing the propionic acids. And here Clostridium, it's helpful for acetoacetyl S-CoA, and it's this acetoacetyl yes, the production of acetone and ethanol. This we need to know the upper product of the cycle, because in this here the lactic acid is directly produced from this pyruvic acid, but in this propionic acid is not directly produced. Before this propionic acid production, we have the succinate product and malic acid. Okay, these 2 things are very important. And here the acetic acid cycle also we having the formic acid. It's a preform. This formic acids, it's helpful. It's producing these some products. And here you can see. Okay. Oh, sorry, I made a mistake. Okay, here Clostridium helpful for the acetic acid production. It's a CO2 and H2 production. Sorry, like E. coli and Salmonella helpful for the CO2 and H2 production. The Clostridium helpful for the acetic acid production. This Clostridium also producing the acetone through the isopropanol through acetone, and butyric acid, butanol. These are all the output of the Clostridium fermentation process. In this pathway, acetyl-CoA, acetyl-CoA, the CoA is produced by the Clostridium. It's a major thing in the pathway. And Klebsiella and Enterobacter bacillus, 3 species are helpful for the 2,3-butadiene, and acetolactic acid production. And ethanol production, this ethanol production, we all know all species are for ethanol, bacillus, lactobacillus, all species can producing the ethanol. Okay, these things are the production pathway. Please remember the microbes and product name. And this is one for as for your the species activity. This lactobacillus can formation of, it's helpful for the formation of alpha galactosidase, beta glucosidase a process. Okay, is a this make a activation of the phosphoketones and tannase. This process hydrolysis of the phenolic and depolymerization. Depolymerization means it's a this polysaccharide get degrade, and the liberation of the phenolic compounds after the hydrolysis process. This is hydrolysis means always breakdown. Ketone compounds need to be liberation of the phenolic compounds, then metal collecting activity, then increasing the radial scavenging activity. It's the fermentation process. They are doing different step by step events. That even that ring is one important step. These are all the products. Okay, these products are helpful for the each and every step. The species is helpful for the product production. Okay, this microbes can produce the produce two type of microorganisms, products. One is the micromolecules, another is the macromolecules. In that, macromolecules means that the minerals, gases, water. The very important thing, micromolecules means it's a 18 to 800 Dalton molecule. This found in the acid soluble pool. Okay, they can acid soluble. Okay, the micromolecules are having small size, low molecular weight, that all know. The 18 to 800 kilodalton, that is very important thing, and it's found in the acid soluble pool. That a large high molecular weight, the more than 10 dalton, the more than 10,000 dalton is called as the macromolecule. Here the found in the acid soluble pool. Okay, it's soluble, was it insoluble pool. The macromolecules are found in acid insoluble pool. Okay, in that these are all the carbohydrate biomolecule, the production and application. Okay, what is the carbohydrate biomolecule means the carbohydrate based which can produced from the microbe, that is helpful for the various application. Okay, first thing here is the compound name is cellulose, and the cellulose is produced by the bacteria called Agrobacterium tumefaciens or the Acetobacter xylenium. This helpful for this product is helpful for skin therapy because it's gives new cell generation. That's artificial blood vessels production, the potential scaffolds of tissues engineering. Scaffolds means it's producing the new organelle parts. So in that case it's a high rich of source, so it's able to make a strong bonding source, so it's able to make a strong bonding with the already available tissues, so it's and also it is easily growable. And the E. coli, it's murine production in the it's a structural protection based one, and agros and agar are producing by the Gracilaria species and Pyropia species. Macroalgae, these are all the macroalgae, and algae having the rich amount of carbohydrates. Okay, that is helpful for many purposes. The agar and agros are also used as a food product in many countries. That's the gelling of product and biological wax. Jellies, we all know the jelly that is made up of also, that is made up of the agarose. The food grade agroses are very widely available in the alginic acid that also produced from macroalgae called the Pheophyces, and fungi, it's a Cleocladium. It's producing the product, the fructooligosaccharides, the fructooligosaccharides are helpful for the functional ingredients. It's are all the applications, overall applications, because they are asking 2 mark, right? So in that 2 mark view, we need to, they may ask the acid and the product and the species, these are all about the product. Okay, product based one. In many apart from this, we have n number of products in the fermentation in a biological subject. Please refer with the materials and some extra things about this the fermentation product. Please refer the individual pathways. Okay, lactic acid production, right? That industrial biotechnology products. That book we have the all the informations about the product and the microbials. Then next, first is the microbial techniques. Then we are going to discuss about the immunological techniques. In this immunological techniques, we are what is the application means? It's diffusion. Diffusion means it's a binding. It's make a collaboration. Okay, how it's helpful for the find the concentration of antibody and antigen. It's compare antigen and it determine the relative purity, how it pure in the sample, and this is diagnosis, serological survey. All those things are very helpful for the this immunological diffusion techniques. This combination of antibody and antigen is a fundamental reactions in the immunology. All experiments. Immunology is explain about the IG, that antibodies, the antibody interaction, antibody and antigen interaction is the whole basic concept. It's also other apart from the previous previous is a basic one. Okay, it's advanced applications, the detection of narcotic drugs and blood bank screening of hepatitis. It's hepatitis virus, because virus having some that is having blood group that having specific antibody against that virus. So using that that antibodies, the addition of antigen, we can find out and screen the blood group, and measurement of growth hormone level and globulin tumor markers in the tumor. They having the different detection techniques, and tracking the leukemia virus, because the leukemia virus count is count maintenance is very important. We can't maintain the count, so for that we having the immunological kit. It's our diagnosis, treatment of peptide ulcer and COVID diagnosis also comes under this immunological testing. In this we having the radial diffusion. That is, radial diffusion is a single diffusion method, and another one is the double diffusion method. That is the in this radial diffusion method, the antibody is put into a gel and antigen is measured by the size of precipitation. In the double diffusion, both antibody and antigen diffuse from well and get the gel medium into the gel medium. So we having the example. It's for before the session, before the example, we need to the procedure. IgG is a major antibody in a human serum. We need to, it's for example, so for the step by process, the 70% of total immunoglobulin pool, we are taken that concentration is set to 1200 mg per the TL, and Ig is react with the human IgG antibody to form a antigen antibody complex. This single diffusion, one compound is fixed. For example, antibody in case is the anti IgG is added. Anti IgG means it's opponent to the the immunoglobulin that is placed in the gel. If you add the IgG, the sample having IgG means that make a interaction and precipitate. And the double diffusion, you can diffuse the antibody and antigen towards, here the antigen, here it's having antibody, both are make a precipitation in one center part. The line of precipitate will form the antibody binding on the antigen. Here you can see the antigens are binded to the antibody. So this precipitation makes the presence of the antibody. Here the double diffusion method, in the single diffusion, just make a zone, clear zone or precipitations. With this double division, we can here they make a interaction. This interaction make a different precipitate. Here you can see the sample. The precipitate one is then on the other language. It's for its live examples only I'm included. Here identity and partial identity non-identical means the positive that PC means positive control. This positive control, it's the antigen and patient serum. Okay. Here they added, here in this cell having the this wells we sample in the well. Okay, wells on the gel. Well, first we need to make a gel, thin gel on the on glass slide. First thing in the immunodiffusion that is available in the readily available in the market. It's this gel. We need to make a gel on the glass, and the gel we need to make a this round circles with a particular distance. In that well, this is the well, right? This is the well. Microbiologist or your biotechnologist may know the well on the gel, because it's a picking of round parts. It's unremoved it. That in that well we need to add the sample. Okay, if you are added the, here you can this is the antigen. Okay, this is the antigen. This is a positive control, because that positive control means that having the blood sample having the antibody against the particular disease. Because we need to know that whether the person for the patients having the disease or not, means for example, it's if that for no. For example, if you have any disease for against we having some antibody, that positive control means readily available, that similar antibody, that is manufactured antibody, it's a positive control you have added here. So the positive antibody, the positive control, it's make a interaction with this antigen. So here the precipitation happens. And here you can see the patient's control. This is the patient's control. The patient control and antigen make a interaction, and so there is a the band stretch. Here you can see the stretch. This is the antibody. Here they added some concentration of antigen. This is low concentration, so it's make a proper link. Here it's a higher concentration, so it's not properly coming. It's make a diffusion. This is a very important image. It's not important. Here you can see this image. The antigen and the positive control make an interaction, and here the single stripe. The stripe look like this. Okay, this like the that precipitation looks like this dark color or white color in the gel. It's make a opalescence nature. Opalescence means that is a cloudy nature on this gel. Here the patients and antigen control, they are providing the image and ask the question, so only I am included this image. Here the other case, the partially identical means it's not fully make a curve, it's partially, so only the it's not uh, fully interacted, it's partially interacted, so only it's called as partially interacted. Here it's crossed one, that is not directed, this antigen and patient is not interacted. Antigen and positive control, that positive control get interacted, so there is no so not identical. In the normal when they that's these are all about the immunodiffusions, double diffusion, single diffusion. In this next concept is the vaccine production. Okay, in the vaccine production, it's first in the history, the Edward Jenner, we all know, he produced this. He is a physician. He produced vaccine for cowpox virus. This cowpox virus vaccine is produced on 1796. It's Latin variola vaccinia. This virus, it's causing the cowpox. So the first vaccine against this virus was produced by the Edward Jenner. And 1885, French microbiologist Louis Pasteur, he and the Emily Roux developed the first vaccine against the rabies. The first invention, the protection is by the Edward Jenner, but for authorized and accepted for all means, that is the French microbiologist Louis Pasteur. We having the Louis Pasteur research laboratory vaccine production in the Uti Kunur area, because one thing that in that particular place we are providing vaccine for all over the world. Why they are selecting that particular place means that is that is a very big history, because the vaccine that we need to control the spread of the research, because it's the viruses can easily spread in any space, and it can easily move to one place to another place. So avoiding this unwanted things they are placed, and we need to maintain at the cool temperature. We want to avoid the denaturation of the antibodies, because the vaccines are mainly focused with the antibodies. The antibodies are they can able to stable in a cold condition. They can't survey that. That antibodies may degrade at the hot temperature. So the place need to be cool. The place should be very protective. So only they are select that top hill area of the Uti, that Kunnur. Okay. That they are selecting, they are searching many countries. After the searching, they are placing the Louis Pasteur Institute and Kunnur. Okay, here the type of the attenuated vaccines. The type of vaccines we are having the live attenuated. This live attenuated example for attenuated means oral polio, MMR, BGC, yellow fever. This is in the killed vaccines. Vaccines are whole cell particle or split vaccine. This is helpful for influenza, the IPV, it's a papillomavirus, hepatitis A, and pet tissues. And subunit vaccine is a meningococcal vaccine and Haemophilus influenza vaccine. Toxoids, it's a diphtheria, tetanus, they are using the toxoid vaccine. Recombinant antigen, it's a hepatitis B vaccine. Combined vaccine that are DTP, MMR, OPV, DTT, the hepatitis B, the Hep B, and KIP. This combined means you may remember in that are your small ages, the doctors are asking us to come the 3 months interval. First one they are providing one drug, one vaccine. After the 3 months they are provided the another vaccine. These are all things are combined vaccines. They are having after the interval some interval they are providing the that's combined vaccines. Here 1 minute. Okay. In this viral vector vaccine, its advantages and here we have different. These are all the activation based types. Here these types are the function that they are compound based one. If there is a viral vector vaccine, it stimulates the innate immunity response. Very important thing. It stimulates the innate immune response. It's induction of T and B cell immune response. Okay, this is maybe the possible questions in the advantage and disadvantage, the disadvantage and advantage for CSIR online their exam. That and it induce the antiviral induction of antivector, its immunity, cell based manufacturing, and the DNA vaccine, it's a non-infectious, it's stimulation of innate immunity. It also stimulate egg and cell free. This is also egg and cell free. There is no egg now, there is cell. Directly they are adding the DNA, and it's a stable, rapid, scalable production, because that DNA incorporated in the human cell that able to produce. And the RNA vaccine, it's non-infectious, non-integrating, because that RNA it's specifically produced from the virus, so not able to producible in a human. Thus viral nature, because the virus having specific nature to produce the RNA in a virus alone. Natural degradation, then it's a natural after RNA vaccine injection that makes some response in human body, and after particular period it automatically degrade. This concern will be instability and low immunogenicity, because it's after the injection, during some small period, it's automatically removed. These are all the examples we discussed in the previous slide. Okay, the techniques, the deduction techniques, because till that I'm okay. Type of vaccines, and that concept is the deduction techniques. The deduction means, in that medical terms, the deduction we have histopathological, serological, molecular technique, and this three techniques are helpful for the medical diagnosis purpose. In this histopathological, we the basics of infected tissue, collect the infected tissue, and we can see it's infected by bacteria or fungi, anything, nemones. And serological checks, it's the interaction of antigen antibody. And molecular techniques, it's a nucleic acid based one. It's a tedious one, it's a long term process. It's why needed to. It's how it is selected based on the sensitivity, specificity, and simplicity. Every detection techniques need to be very simple, specific, and sensitive. For example, like nowadays with the coronavirus detection kit, the sensitivity is important. You may hear the news, it's at 52% sensitivity, it's 82% sensitivity from China. That various percentage they mention, the percentage is the sensitivity. That sensitivity, the normal, in the normal day, normal conditions, the detection technique system, it's allow the 99% sensitivity. For the pandemic period, they are allowing some detection kits. Here the specificity is very important. Sensitivity, okay, the sensitivity, it's want to be too sensitive. It can detect a very small amount of target means that sample. Then specificity and simplicity. Here some other microscopic methods in the diagnosis, that is microscopical examination, in vitro, and mouse inoculation. After the injection into mouse, and we can detect the mouse image on the sensors. Detection of the antibody in the serum, and DNA hydration, and PCR methods. These are all the techniques. Okay, with the microscopic we can see the cell structure. If there is a if you want to check any drug, means the drug effect, that check by diagnosis by the animal, that mouse. An antibody and detection, as I told, the that is the for example, that is a COVID detection kit. And DNA hybridization and PCR, it's helpful for the protein finding. The hormonal test, or any DNA based DNA lagging, if there is any mutation in one cell, if there is a cancer patients, we need to understand which gene is affected. All those things are diagnosis using the DNA hybridization and PCR. And the diagnosis method and the interpretation, how we can interpret. Here these are all the steps: microscopic, gram staining, phenotype, we can growth, we can find the growth, in vitro antimicrobial assay, we can antibiotic selection we need to do. Then real-time PCR. Real-time PCR is also legendary one for the any antibody selection, antibody detection. Mass spectroscopy is helpful for the diagnosis of the compounds. PCR is a polymerase chain reaction. You may all hear that word in the TV also, because this RT-PCR, there's a real-time PCR, is helpful for the detection of the our products. Okay, next the important concept is the tissue culturing. In this tissue culture, we having the different two types. The one is plant tissue culture, another one is animal tissue culture. In this plant tissue culture itself, we having the different methods. One is type of in vitro callus suspension growth and explant growth. We can grow the whole plant with single cell. This using this single cell, we can produce the whole cell, right? That concept is or a single cell culture, shoot and root culture, somatic embryo culture, meristem culture. These are all the some types. Okay, these are all the methods. And the protoplast culture, somatic hybridization, embryo culture, the oval culture, ovaries culture. These are all the explant cultures. Here in animal cell culture, we having two types, that is the primary culture and second continuous culture. In the primary culture, we having we can grow the that excised tissue in a culture. Outgrowth, it's just a outgrowth from the single cell, okay, derived directly from the existing tissues. And in this continuous culture, it's a subculture after the culture of the primary culture cultivation. That is the after the step of primary culture, we want to take that primary culture and cultivate in another medium, fresh medium, that is the subculture. This continuous culture, we having two type of cultures, cell lines and continuous cell lines. It's a type means in this is a this types are the based on the cell. We can grow the stem cell, we can grow the precursor cell, we can grow the differentiated cell. For stem cell, we can produce what you need. For that you need, if you want a heart, if you want a liver, we can grow whatever we want. In this differentiated cell, that culture that usually do not capable to divide, so we can't divide into for the cultivation. And precursor, they divided into derived from the stem cell. It's committed to differentiation, but are not yet to differentiate. It's retain capacity for the reproliferation. In plant tissue culture, we are having steps. First, we want to collect the plant and add the disinfectant to remove the other extra tissues, extra dust on the plant, and remove the microbes on the plant. Then grow the callus. After taken the callus, that placed on the medium, the specific medium. That specific medium is contains the root forming material. That callus make a root, and after the root formation, that root formed culture, that tissue, the plant is replaced in the shoot forming medium. That shoot forming medium helpful for the shoot formation. That formed plant is placed in a pot. These are the stages. First, we need to cut the soft wood. That soft wood, it's helpful for the production. After 2 to 3 months, it's make a shoot. Its shoots are established. In the cycle development, its cluster of roots are there. Then that after the stage, that is the first here the first stage is a shoot manipulation. Then rooting, that roots are formed, individual shoots are placed in a medium, and here they are forming the root. That root formed plants are acclimatations on the gel can or sealable, that sealable plant. This is the overall sealable plant, and they are replacing this in a mud contained pot. Some enzymes are used in the medium. These enzymes are some compounds, not enzymes. These compounds are helpful for the whole process. These are only some compounds, because they are all must for their production. The production, that means shoot production, root production. We all know the gibberellin, and for their root, we need to add some hormones also. In the nitrogen, potassium, calcium, magnesium, phosphorus, sulfur. These are all the major compounds, major elements in the plant tissue culture. And these compounds are helpful for various. Please refer this table for your understanding. And in chlorine, the ions magnesius and cobalt, copper, zinc, molybdenum. These are some other cofactors. Here till this step I mind up, because after that we having the plant tissue culture and some other methods. Today we are discussing the techniques, fermentation process, what is immunology related techniques, detection techniques, and the vaccine production, the vaccine types, and the next we are discussed the plant tissue culture. Tomorrow class we will discuss the remaining part of the unit. 12. Tomorrow is the last day of our online 30-day training, because that is the 30-day planned one. Till tomorrow I will finishing the unit 12, that is the third session. Thank you everyone. If you have any queries, please ask in chat box. In human system, we having 5 type of antibodies, ma'am. IgG, that IgG, IgM, IgE, IgA, IgB. That 5 things. Apart from this antibodies, these are all the 5 major antibodies, because immunoglobulins is a antibody system, it's a representatives. It's apart from that different antibodies, because for diarrhea we need to develop for antibody. For diseases, for various diseases, we having various antibodies, different system. From the childhood to the age, at the old age, we having 10 more than 10 billion different human system. Major thing is the IgG, IgM, IgA, IgE and IgD. These are all the major immunoglobulin types. These are generating the generating that this major immunoglobulins are generating the different antibodies for their that modulating the differentiate into different antibodies for their various attack. Okay, I think there is no questions. Now I am going to end this streaming. Thank you everyone.